to Europe. We also have a lot of super isolation, yes. My last flight was 1 year ago. Can you imagine that? I don't hardly know where the airport gone to. I really have to find out. Hopefully this comes in the next few months.
It must be very difficult for you, Manfred. You must.
Oh, it's a disaster.
We are live, sir.
We are live. Perfect. Okay, so welcome, ladies and gentlemen, to our 6th webinar. We finished our 5 basic uveitis courses already. Now we're going a little bit more into detail. So you'll have much more information because you've learned already your first 5 lectures. If not, if you missed some, you know where you find the whole videos. They are where you register. Scrolling down there, you will find it. Few words. After registration, participating in that course here, you will get a evaluation sheet. Very important to send that back because only with this evaluation sheet, you will get your documents and the CME points. And we need this evaluation sheet too, of course. There is a sign, question and answers. Please use this. We will try to do our best to answer as soon as possible. Do not use the chat button. This is actually only for communicating for us, actually. Use the question and answer button, but only to questions which are related to this topic, please. Otherwise, uveitis is such a big field, we cannot go too much over this one. Afterwards, we will stay online a little bit after these 2 hours and try to finish all the questions you have sent. Again, if you missed something, you will be able to see that on our website. Okay, so with this, I think we could start. And I think, Dennis, can you start with anterior uveitis genetics and epidemiology?
Manfred, you've disabled the participants share screening.
Yes.
I need to share the screen. Host has disabled participants share screen sharing. I can't.
You are allowed to share, please.
I think it's okay to click only on your presentation. That should open everything.
Dennis, the best thing is keep your presentation open.
Okay. Can you see my presentation now?
Yes.
Yep. Go to slideshow. Perfect.
Thank you.
Go on.
Thank you very much. I'd like to discuss this evening or today some of the epidemiological and genetic studies that have been carried out in anterior uveitis that have given us insight into the pathogenesis of this disease. I'll summarize some of the major factors that are important from a point of view of understanding the types of anterior uveitis that occur in different communities across the world. Our basic understanding of the pathogenesis of uveitis is that there are 3 main factors involved. These include genetic factors, which I'll concentrate on today, environmental factors, which consist of microbes and, of course, the microbiome, which again is one of the topics we'll discuss today. Drugs have been in the news in recent times, especially with the checkpoint inhibitors causing uveitis, as well as now a large list of other drugs that may trigger uveitis. And of course, the immune response, which contributes to the inflammatory process and also localizing events that, for reasons that we still don't understand, chooses the anterior uveitis as a target for the inflammatory response. If you look at the prevalence of uveitis across different countries of the world, as shown in this slide, with each of the major countries, and I've been selective in choosing these countries, it's mainly because of the size of the studies, the populations that were studied as part of this. And just about every country that one looks at, anterior uveitis is the commonest form of uveitis. Now, most of these studies actually were carried out in large uveitis clinics, and therefore, in a way, are prejudiced towards more severe forms of uveitis, such as the posterior uveitis and panuveitis. However, even within those, sorry about that, even within this group of patients, you'll notice that anterior uveitis is the most common form of uveitis in just about every country in the world, and HLA-B27 anterior uveitis in particular is the most common subtype of anterior uveitis. In community-based studies, it is estimated that up to 70% of all cases of uveitis are anterior uveitis, and about 40 to 50% of these people, depending on the part of the world you come from, will be HLA-B27 positive. If one looks at some of the commonest causes of non-infectious anterior uveitis, and this is not meant to be a comprehensive list, but to point out the importance of HLA-B27 uveitis and spondyloarthritis in different parts of the world. And you'll notice again that consistently there's a high prevalence of HLA-B27. Similarly, Fuchs is reasonably not common, but it's prevalent in all the major parts of North and South America, Europe, India, and Asia, as well as in Australia, as is JIA. But again, the most prevalent form of anterior uveitis is that associated with HLA-B27. And amongst the people who have anterior uveitis, spondyloarthritis is the commonest disease association, particularly associated with HLA-B27. It would be remiss of me if I didn't mention some of the other genetic causes of uveitis. And in fact, there are a large number of autoinflammatory diseases which are monogenetic that are associated with uveitis. The interesting thing about these diseases, although they are all quite uncommon or rare, a large number of them are actually now known to be due to abnormalities in the inflammasome pathways. They also may involve the immune system in terms of the NFK beta dysregulation, macrophage activation. Well, in the case of the TRAP syndrome, they're believed to be triggered by innate immune systems, with pathogen-associated molecular patterns or DAMPs being important. If we look at the HLA associations that have been reported in uveitis, the increasing number of subtypes of uveitis are associated with these genetic risk factors. In terms of anterior uveitis, which I've highlighted here in red, acute anterior uveitis is associated with HLA-B27, has a high relative risk, and is quite prevalent. We also know that people who are homozygous for the HLA-B27 gene have a 3 times increased risk of developing uveitis compared to people who are heterozygote. Behçet syndrome and TINU also associated with genetic factors. In the case of Behçet syndrome, HLA-B51 is prevalent in most of the countries in which this has been studied, as similarly with tubulointerstitial nephritis and the uveitis syndrome. What's interesting about HLA-B27, when this was first discovered, which is almost 50 years ago now, we thought this was the answer to the puzzle as to why there was genetic concordance with B27 and uveitis. But as time has gone by, we've now realized that HLA-B27 consists of about 170 subtypes. And not all of these subtypes are, in fact, associated with uveitis or with ankylosing spondylitis, as shown in this slide. The most common subtype of HLA-B27 associated with uveitis and the spondyloarthritis or ankylosing spondylitis type diseases is the HLA-B2705, but it also occurs in people that are B2702 and B2703 and 04. Other subtypes, which there are obviously a lot, aren't associated with angspond or with anterior uveitis. In most urocorticoid populations, the B2705 is the most prevalent subtype of HLA-B27. We've recently been involved in a large genome-wide study of patients with anterior uveitis. This involved over 1,700 patients with ankylosing spondylitis and uveitis, or just HLA-B27 anterior uveitis. And the results of this study reveal, in fact, there are more genes involved in the pathogenesis of these diseases than was previously suspected. Obviously, B27 was the standout, and as I've already mentioned, there are over 170 subtypes of B27 now recognized. But also several other genes were found to be or reach clinical significance in terms of their association with uveitis. These include the ARAP1 gene, which is shared with ankylosing spondylitis, the MERK-K gene, which is also associated with ANKSPOND, as is the interleukin-23 receptor and IL-6 receptor genes, and an intergeneric region called TP15. The other genes outlined in this table were increased, but did not reach gene-wide significance. So it's now apparent that ankylosing spondylitis and anterior uveitis, particularly those associated with B27, are in fact polygenic, and there are more genes involved than was originally suspected. The ARAP1 and 2 genes, these are the endoplasmic reticulum aminopeptidases, are important in trimming the peptides before they're loaded onto the HLA class 1 genes. HLA usually binds peptides 8 to 10 amino acids in length, and these peptides are trimmed by the ARAP1 and ARAP gene so that they actually fit into the groove on the HLA-B27 molecule. These proteases are also involved in cleaving cell surface cytokine receptors, which obviously would be important in immune responses. And polymorphisms of the ARAP gene influence the peptidome of the B27 molecule. So that, in other words, they alter the epitope of the peptide that binds to HLA-B27. They have other functions as shown in this small slide in the corner here that in fact indicate that these have a role in blood pressure control, angiogenesis, as well as antigen processing and macrophage function. And it may be, in fact, that by targeting these sort of protease genes, there may be another avenue for not only understanding the pathogenesis of these diseases, but also for therapeutic intervention. The MERK-K peptide is a member of the receptor kinase family, it's a transmembrane protein, and mutations in this have been associated with retinitis pigmentosus, as is the EYS gene. High levels of this kinase have been found in the retina and expressed in macrophages, dendritic cells, NK and NKT cells, which are obviously important components of the immune response and have a role in suppressor T cell responses as well. They also have a role in clearing apoptotic cells and reorganization and engulfment of material by the immune system and macrophages. What their role, however, is in autoimmune disease and in the pathogenesis of uveitis has not been ascertained. So in terms of take-home messages with regards to anterior uveitis and genetics, it's now apparent that there are a large number of HLA-B27 subtypes, of which only a few are associated with anterior uveitis and with ANKSPOND as well. And that anterior uveitis and spondyloarthritis are polygenic, which have strong shared gene interactions, as I've just outlined. Anterior uveitis is commonly associated with systemic diseases and may in fact be the initial manifestation of one of these spondyloarthropathies, and we'll come back and talk about this in a little while. These genetic factors may also affect the microbiome, and Jim Rosenbaum is going to talk a bit more about this. And it's been proposed that the discoveries of these new gene associations with the HLA-B27 cluster of diseases may lead to new insights, not into pathogenesis, but provide new avenues for therapy based on these genetic studies. So thank you for your attention.
Thank you, Dennis, for a very amazing opening. And now we will go on to Peter. Peter is going to go on to the clinical presentations, features, and grading of HLA-B27. Dennis, you have to go right on the top and stop sharing.
Thank you.
Great.
Great.
Vishali, can you see my screen?
Absolutely. Yeah.
Great. Okay, well, look, hello, everyone. It's a real pleasure to be talking to you today and tonight. My first talk is going to be about clinical features and grading of B27-associated anterior uveitis. And there'll be some overlap with the other talks, and that's actually a good thing because it will just reinforce some of the important messages that we want to get across. As Dennis said, anterior uveitis is the commonest form of uveitis. It's it's around 50% of all the patients that we look after with uveitis. It's defined as inflammation that involves the iris and ciliary body and is centered obviously on the anterior uveal tract and therefore called anterior uveitis. About up to 75% is episodic and recurrent. That is, it's acute anterior uveitis. You can also get chronic persistent anterior uveitis, and there's a wide range of causes, both inflammatory and infectious. Again, as was mentioned earlier, around half the patients that we see with acute anterior uveitis are HLA-B27 positive. It's probably more common in males than females, but the predisposition varies a little bit depending on how you look at the disease. And again, as Dennis said, at least 50 to 70% of patients who are B27 positive have a systemic B27 related disease. The little cartoon up here on the top, on my top right, basically shows that acute anterior uveitis can be associated with all of the different spondyloarthritises and systemic B27 related diseases, but overwhelmingly ankylosing spondylitis is the commonest. It's an axial spondyloarthropathy, but you do get B27 related AAU in patients with psoriatic arthritis, with reactive disease, and with inflammatory bowel disease. We know that anterior uveitis, the risk becomes higher with increased duration of your spondyloarthritis. And there's a very good evaluation tool called DUIT, the Dublin Uveitis Evaluation Tool, which is a good way of just sort of analysing patients that you see with acute anterior uveitis. I won't go into it tonight. It's easily findable in our literature and is a really useful little algorithm to use in the clinic. We know that, as I said, that the duration of your of your spondyloarthritis increases your risk of developing AAU, and this is a large study that shows that gradually over time you do get more frequent acute anterior uveitis. It does vary, as Dennis said, around the world, but it's common no matter where you're practicing. So the clinical features of acute anterior uveitis are characteristically that it has a sudden onset, that it is typically unilateral. Obviously, acute anterior uveitis can be bilateral, but B27-related acute anterior uveitis is unilateral and it alternates between eyes. It has a high frequency of recurrence. It has episodes that are variable in their severity, and very importantly, it is of limited duration. And most patients do very well with treatment. These are the sort of clinical signs you see: red eye, light sensitive, photophobic, small pupil. Sometimes it can be very severe, as you can see with this fibrin veil here in the image down the bottom. We're trying to use the SUN nomenclature that many of us have been involved in, and classically HLA-B27 associated AAU has sudden onset, limited duration, an acute course, and it can be recurrent. And we want to try and use this language as our descriptors when we're describing an episode of uveitis, no matter what phenotype it is. If we just sort of hone down a little more on the clinical features, as I mentioned, these patients typically present with a red, painful, photophobic eye and blurred vision. if our patients have recurrent disease, they can usually tell a day or two in advance before we can see physical signs that they're getting an attack of anterior uveitis. These episodes are very stereotypical in their presentation, and our patients know that they're getting an episode. We obviously see flare and cells in the anterior chamber. We can typically see these very fine, almost dust-like KPs in the inferior cornea. If it's very severe, you can get fibrin, synechiae, a hypopion, as you can see here. The intraocular pressure is classically a little low, but it can be normal. It usually doesn't go up immediately, but you can also, of course, get elevated intraocular pressure. And critically importantly, the fundus has to be normal. I've mentioned that you see cells and flare, and you can see the little white dots that are macrophages and polymorphs in the anterior chamber, and the flare for protein leaking from the vessels. And there's sun grading schemes that we use to sort of grade the severity of the flare and the cellular response. As I mentioned earlier, acute anterior uveitis, particularly B27 AAU, can spill over into the vitreous. When it does this, it's in the anterior vitreous. It's not a pan-vitreous cellular infiltrate, and the anterior chamber cells are much more severe than the vitreous cells. B27 AAU can evolve into chronic anterior uveitis if you are unable to control the episodes. You can get more severe vitreous involvement. Certainly posterior uveitis has been documented in the literature, and we'll talk about that a little more later. Scleritis is an increasingly recognized association of HLA-B27 and spondyloarthritis, but is clearly separate to B27 AAU. And now that we've got Optos wide field digital imaging and angiography, we not uncommonly see some mild peripheral leak and non-occlusive changes in the far retinal periphery. The exact significance of those is not quite clear at the moment. Importantly, you can lose vision from anterior uveitis, and the important clinical sort of thing to do is to work out, is it inflammatory mediated, where it's classically cystoid macular edema in AAU, or is it non-inflammatory related to a cataract or elevated pressure, band keratopathy, occasionally to hypotony. Obviously, non-inflammatory vision loss won't respond to anti-inflammatory therapy. So to summarize, we know that there is this strong relationship between acute anterior uveitis and recurrent anterior uveitis with the spondyloarthritides, particularly with ankylosing spondylitis. We know that AAU is a risk factor for ankylosing spondylitis, and we importantly know clinically that 90% of our patients get either acute anterior uveitis or recurrent episodes of acute anterior uveitis that alternate from eye to eye. Only small numbers of our patients get chronic anterior uveitis, and the sort of standout group is probably the reactive arthritis group. And as I mentioned earlier, patients who've had very severe episodes of recurrent anterior uveitis can develop chronic persistent anterior uveitis. Intermediate uveitis and scleritis are reported in association with spondyloarthritis, but they're uncommon, and it's very, very uncommon to get posterior uveitis with spondyloarthritis. The major risk factors for relapse of acute anterior uveitis are a younger age of onset, and obviously being B27 positive and having a spondyloarthritis. And as you can see from this large study, the risk of a relapse approaches 40% by 18 months after your initial episode. just recently in the American Journal of Ophthalmology, all the Sun classification criteria for 25 phenotypes of uveitis have been published. And these are the classification criteria for spondyloarthritis associated B27 associated acute anterior uveitis. And as you can see, you've got to obviously have anterior uveitis. You need to have that characteristic clinical course of acute or recurrent acute. It can be unilateral, but it classically is unilateral alternating. As I mentioned earlier, you can have chronic disease that's evolved from acute recurrent anterior uveitis, and obviously you need to have a spondyloarthritis if it's a spondyloarthritis associated acute anterior uveitis. Manfred's going to talk about these diseases down here, the exclusions, syphilis and sarcoid and viral anterior uveitis. So I won't dwell on those here, but they are the important diseases to rule out in our patients with acute anterior uveitis. So I'll stop there and hand back over to Vaishali.
Thank you. Thank you, Peter. That was very useful. Peter and Dennis, there are a few questions for you in the Q&A box, which you can start answering whenever you get time between the talks.
No problem.
Move on to Dennis again, who's going to talk about systemic disease and anterior uveitis. Over to you, Dennis.
Thank you, Vaishali. Can you see my screen there?
Not yet, Peter.
Can you see my screen, Vaishali?
No.
Oh. Excuse me for 1 second.
Rohit, is Dr. Dennis able to share? Have you given him?
Okay, thank you.
Yes.
Yeah.
Can you see my screen now?
Perfect. Go to slideshow and you're all set.
Okay, thank you, Vaishali. So I'm going to discuss some of the systemic diseases associated with anterior uveitis. And Peter's already highlighted some of the major diseases that are associated with acute anterior uveitis. Just for completeness sake, I thought it's worthwhile reflecting on the causes of anterior uveitis. And there's a handy little mnemonic for those that don't see a lot of uveitis that I think is helpful in reminding us of what the causes of anterior uveitis are. And that's the mnemonic iritis, where I stands for infections, R for rheumatic diseases such as the seronegative arthritis, Behçet syndrome, juvenile arthritis, infiltrative diseases, which brings up the whole concept of masquerade syndromes in uveitis, which is something that we need to be aware of and on the lookout for, trauma causing sympathetic, immune diseases such as TINU, inflammatory bowel disease, lupus, and rheumatoid arthritis rarely, and systemic diseases such as sarcoidosis. In terms of how one approaches the detection of systemic diseases associated with anterior uveitis, I'll use this 5S approach, if you like, which again is a sort of a useful mnemonic for how to approach detecting a systemic disease in a person presenting with anterior uveitis. Probably the most important thing is to take a careful history and careful examination, both an ocular examination and a systemic examination when it's indicated. Symptoms and signs, as Peter outlined already for anterior uveitis, are obviously important, but so are the features that typically give a clue to there may be a systemic disease. Obviously, you need to do a careful systemic review looking for these other illnesses. Often this is detected by recognizing a particular syndrome, and we'll talk a little bit more about that. And finally, selective investigations. And that I'll emphasize how important selective investigations are. It's no use doing every test known to mankind because they're usually a waste of time, energy, and money. So in terms of systemic reviews, again, The things to concentrate on are whether the person has skin disease, synovial involvement, serosal involvement, mainly of the oropharynx and gastrointestinal tract, and whether the person has systemic symptoms. In terms of the skin disease, of course, psoriasis is common, as is erythema nodosum, as is ulcers, and a number of other features which should give a clue to the fact that there may be an underlying infection, inflammatory disease, Behçet's syndrome, or psoriatic arthritis. Synovium is the clue to carefully look for evidence of arthritis, especially spondyloarthritis, and I'll come back and talk a little bit more about that. Serosal surfaces with mouth ulcers and genital ulcers in Behçet's syndrome, but also these occur in people with inflammatory bowel disease as well. And systemic symptoms, night sweats, fevers, lymphadenopathy, etc., should alert you to the fact that there may be another more sinister disease underlying the person's uveitis. Here's a person with a cluster of diseases which would be easily detected on careful history and examination, with multiple aphthous type ulcers, genital ulcers, and anterior uveitis with hypopyon, and erythema nodosum, a form of paniculitis on the legs. And this is a person with the classic features of Behçet's syndrome. Synovitis and the detection of inflammatory arthritis is probably one of the most important aspects in assessing the presence of a systemic disease associated with uveitis. The major feature of this is the detection of inflammatory back pain. And inflammatory back pain has a number of typical features. These include pain and stiffness, which is worse after rest and lasts for more than 3 months. This contrasts with other degenerative types of arthritis, osteoarthritis, etc., which are worse after activity, but of course can be persistent as well. The pain in inflammatory arthritis often improves with activity and is also characterized by nocturnal pain, people waking up in the morning with stiffness or waking through the night with joint pain. In the seronegative arthritis or the spondyloarthropathies, there's often lower limb oligoarthritis. That's mean one or more joints are involved. And another classic feature of this is so-called enthesitis. The enthesis is that connective tissue of the muscle that inserts into bone, and people with these diseases, these seronegative arthritis or these spondyloarthropathies, often have inflammation of the enthesis around the pelvis, around the knees and ankles. And dactylitis refers to involvement of the small joints, usually of the hands and feet. It's important also to look for evidence of psoriasis and inflammatory bowel disease. These diseases are more typically more common in men, although more recent studies have shown that often females will have a milder form of spondyloarthritis that goes undetected. They usually have their onset in young people less than 30 years of age. We now recognize, in fact, that there are a group of spondyloarthropathies or a family of these diseases, including classical ankylosing spondylitis, the so-called undifferentiated spondyloarthropathies, reactive arthritis of Reiter syndrome. There's a juvenile form of spondyloarthritis, as well as arthritis associated with inflammatory bowel disease and psoriatic arthritis, as we've already mentioned. These diseases are all associated with B27. In this slide, you can see a person's got Achilles tendonitis, another form of enthesitis, oligoarthritis with swelling around the knee, and the X-ray of the spine shows something you don't want to see, which is actually the presence of syndesmoid phytes and ankylosis of the spine. One of the big advances that's been made in terms of detection of this group of diseases is the use of MRI, which not only can detect early signs of sacroiliitis, but also detection of changes in the adjacent bone marrow, which are typical, in fact, of sacroiliitis and are helpful in the early diagnosis of this disease. Psoriatic arthritis is associated with uveitis. Peter's already summarized the major features of this. It's sudden onset, unilateral. 77% of people with psoriatic arthritis will develop uveitis, and 4% of psoriasis occurs in people with anterior uveitis. The arthritis often precedes the uveitis in this group of diseases. And as Peter's already pointed out, they're non-granulomatous with fibrin, iris bombay, and often hypotony. Reactive arthritis also is something that's classically associated with this family of B27-related diseases. This disease is usually has its onset 1 to 4 weeks after an infection, usually of the genitourinary system, and this person's got a urethritis, or of the gastrointestinal system. There's a sudden onset of anterior uveitis. It's again unilateral. It classically is associated, or the very severe forms of it in particular, are associated with HIV infection. And anterior uveitis is present in about 40% of patients who have Reiter syndrome. And in Reiter syndrome, 9% of them have B27 anterior uveitis. Overall, however, the prevalence of B27 is quite high in people with reactive arthritis and uveitis. In other words, with Reiter syndrome and anterior uveitis. Again, it's non-granulomatous, AC and fibrin as well. Inflammatory bowel disease, and you can see in this X-ray, this person's got a very smooth colon due to extensive ulceration of the colon, and this gastroscopic view, endoscopic view shows areas of ulceration of the mucosa on colonoscopy. Inflammatory bowel disease is detected in about 2 to 12% of people with anterior uveitis. It can have other features, ocular inflammation besides anterior uveitis. It's more common in Crohn's disease than in ulcerative colitis, and anterior uveitis is more likely associated with inflammatory bowel disease, where there's an associated spondyloarthritis. This, in contrast to other forms of spondyloarthropathies and uveitis, this is more common in women, and often this occurs early in the course of the disease. I mentioned before about the importance of syndrome recognition in detecting associated systemic diseases with uveitis, and we've already mentioned most of the systemic diseases, the spondyloarthropathies, Behçet's, juvenile arthritis, inflammatory bowel disease, and TINU, and syndromes such as Blau syndrome, but also ocular disease where B27 anterior uveitis may occur on its own. Sorry about that. Without associated systemic disease, as may sympathetic ophthalmia, Fuchs and Cogan syndrome. One of the diseases that's uncommon but has a classical presentation is the tubular interstitial nephritis syndrome, which is in fact more of a systemic disease. It's not common and occurs in about 1 to 2% of cases of uveitis in most clinics. And 10% of the cases present with sudden onset of bilateral anterior uveitis. And when this occurs in a young person, one should then be aware that we should look at the urine to see if there's evidence of an active sediment that would indicate the presence of an underlying renal disease. It usually responds to local therapy, although some people require systemic steroids and other therapy, as we'll discuss later. And there's usually a good prognosis. It also has a genetic association with the HLA-D-QR phenotype or genotype. Let me just say a little bit about selective investigations looking for evidence of associated systemic disease. The aims of these investigations should be to substantiate the provisional diagnosis of the ocular and systemic disease, to exclude relevant differential diagnosis, establish the severity and complications of the disease, and ascertain prognostic factors and complications that may emerge from the treatment, particularly systemic immunosuppression, which we'll talk about in a little while. And there should be a tailored approach to these investigations. In terms of selective investigations, there's potentially a large number of tests that may be indicated in patients with a variety of different types of uveitis. By far, the most important test to do in a patient with uveitis is to take a careful history and physical examination. The other tests will depend on your provisional diagnosis and differential diagnosis, and also looking for problems that may emerge if you're going to immunosuppress the patient. I'd like to emphasize, however, that extensive unfocused laboratory screening test is inefficient, costly, and may not contribute to the underlying diagnosis. A few years ago, the group of people published their recommendations for investigations in people with anterior uveitis, which I think are reasonably sensible and still applicable now. Non-granulomatous anterior uveitis with a first attack that's mild probably doesn't require treatment, but does require a careful follow-up and information to the patient about possible other diseases. If there's a severe first attack, it's probably worth doing their B27. And in our clinic, we would do that simply because we're interested in this genetic factor. Recurrent or chronic severe non-granulomatous disease requires further investigations with chest X-ray, VDRL, and B27. Granulomatous anterior uveitis, raising the spectra of TB and sarcoid, does require further investigations with chest X-ray, VDRL, looking for evidence of TB and sarcoid with PPD and ACE, and in people in which it may be relevant to do a gallium, or we'd now do a PET CT scan looking for this. And if there's any tissue that could be biopsied to substantiate the diagnosis of sarcoid, then we would carry this out. An AU in a child, that's important obviously to look for the presence of ANAs and B27. So in terms of systemic diseases with uveitis, I think there are a few take-home messages. First of all, to emphasize that a significant number of patients will have an undiagnosed systemic disease at presentations with their anterior uveitis. It's important to have a systematic approach to looking for these systemic diseases, and I've indicated how we do it, this 4S approach. Syndrome recognition also will be helpful in about a quarter of the patients we see. If further investigations are required, these should be selective and focused on arriving at a diagnosis and looking for factors that may be important if you're going to further treat people with systemic therapy and immunosuppression. And I think it's important to also to emphasize that regular review and reflection are quite rewarding when you follow up patients with acute and recurrent or chronic anterior uveitis. Thank you.
Thank you, Dennis, for highlighting the importance of systemic investigation and when should we order. Thank you. With this, we move on to our next talk on microbiome in anterior uveitis. It is by Jim Rosenbaum, and since he's on the Pacific Coast, which is midnight for him, we have a pre-recorded talk. But still, we'll be happy to answer any answers that you might have after the talk. So Rohit, can you play Jim's talk, please?
My name is Jim Rosenbaum, and I'm going to try to explain why I think the microbiome may be a cause of uveitis. This lecture is dedicated to Mark Asquith. Mark and I worked together for about 7 years, and Mark passed away 2 years ago from a cerebral aneurysm. So I'm going to begin by describing a puzzle, and then I'm going to seg away from that puzzle to describe the microbiome, review the concept that bacterial products might be a cause of uveitis, and then see if I can juxtapose the concept of the microbiome and the puzzle that I've described. I had the good fortune of training more than 40 years ago with Hugh McDevitt. And Hugh McDevitt is the father of the concept that immune response genes govern the immune response. He did this by taking short repeating peptide sequences, injecting them into mice and rabbits, observing that certain animals made an immune response and others did not make an immune response, and correlating the ability to make an immune response with the major histocompatibility complex. McDevitt and Benasareff, who got the Nobel Prize, wrote a seminal editorial in the journal Science almost 50 years ago. They said, this type of genetic control of a specific immune response may play an important role in susceptibility to a variety of diseases in both animals and man. And it was only a few months after that editorial was published that it was actually validated. And it was validated in a surprising way. It was validated in a disease called ankylosing spondylitis, which many rheumatologists believed was not really an inflammatory disease. but a group in London and a group in UCLA almost simultaneously reported that ankylosing spondylitis was associated with an HLA molecule that was then called HLA-A27, and we subsequently know it as HLA-B27. Approximately 90% of everyone who develops ankylosing spondylitis possesses the molecule HLA-B27, and it turns out that acute anterior uveitis, which we know to be strongly associated with ankylosing spondylitis, is also affected by the HLA-B27 molecule, and that was also published by this group from London, led by Derek Brewerton. So in 2021, we know that HLA molecules affect susceptibility to more than 100 diseases. Almost all of those diseases are immune mediated, and we don't know why. We know that HLA controls the immune response, but we don't really know the true linkage as to how HLA molecules might affect susceptibility to those diseases. So let's leave the concept or leave the puzzle and go to the microbiome. Bacteria in our body outnumber the cells in our body. And if we look at it on a transcript level, on an RNA level, for every one transcript coded by a mammalian gene, there are probably 100 or more transcripts coded by a bacterial gene. And that doesn't include the transcripts that are encoded by viruses or helminths or archaea that are also present in the body. Until about 2007, we knew very little about the microbial world. But in 2007, the cost of DNA sequencing had fallen enough that it became practical to begin to characterize that microbial world by sequencing DNA and RNA without resorting to cultures. Bacteria are essential for life. We have a symbiotic relationship with the bacteria, which are especially present in our gut. For example, those bacteria are the major source of vitamin K, and those bacteria educate our immune system. The Human Microbiome Project began in 2007, funded by the National Institutes of Health, and a similar project called MetaHit began in Europe about the same time. My colleagues and I got interested in the concept of the microbiome as a possible cause of ankylosing spondylitis and HLA-B27 related diseases about a decade ago. And we wrote a summary of this in the leading rheumatology journal in the US, Arthritis and Rheumatism, now called Arthritis and Rheumatology. My own interest was sparked by an article that was published in Science in 2010. In this article, the investigators examined metabolic syndrome in mice that were lacking an important part of the innate immune system called TOL5. These animals without TOL5 had a change in their gut microbiome. They became obese and they became insulin resistant. But the reason that this paper was in Science is because the metabolic syndrome was contagious. When the mice lacking TOL5 were co-housed with mice that were identical, except they had a normal TOL5, the identical mice, the cousins, the congeneic mice, also developed metabolic syndrome. And that's because the congeneic mice ingested the stool of the TOL5 knockout mice, and that stool transmitted the microbiome and made them insulin resistant. So they showed that the microbiome was a vital component in mice, at least, in maturity onset diabetes metabolic syndrome. So I don't have time to go over in detail all the evidence that the microbiome educates the immune system. But if you raise mice in a germ-free facility so that their gut never gets colonized with bacteria, they have an impaired immune system with shriveled mesenteric lymph nodes, a shriveled spleen, and an impaired ability to mount an immune response. That interaction with the bacteria in the gut is absolutely essential for the immune system to be educated and for the immune system to develop. So let's move along to the third point of the lecture, and that is let's interrogate the question, do microorganisms and innate immunity play a role in uveitis? Well, this concept is more than 100 years old. A.C. Woods was the first head of the Wilmer Eye Institute, which is at Johns Hopkins University. And A.C. Woods would take bacteria, at least supernatant from bacteria that had been grown in broth, and inject that supernatant into rabbits. And when he injected that supernatant intravenously, he said poisons could be isolated from non-pathogenic ferment-producing bacteria, which have a definite action upon the uveal tract in the rabbit eye. Histologically, the eye showed a round cell infiltrate, meaning a leukocyte infiltrate, about the ciliary processes. He observed uveitis from just injecting intravenously the supernatant of bacterial broth. This concept that bacterial products might be a cause of inflammation, not only in uveitis, but in rheumatoid arthritis and in other diseases, had a profound effect on our way that we approached inflammation. So here's a paper from the American Journal of Ophthalmology in 1932. It came from a group at the Mayo Clinic in Rochester, Minnesota, and it was based on a presentation at the second ARVO meeting, which had been held in Philadelphia in 1931. It summarized how the Mayo Clinic in that era approached uveitis. They looked for foci of infection, and they found different foci, including teeth, tonsils, prostate, or cervix. And if you were referred to the Mayo Clinic for uveitis in that era, the treatment was to remove these foci, meaning that a man might have a prostatectomy and a woman might have a hysterectomy to try to eliminate the cause or the purported cause of uveitis. Now, I also, early on in my career, was very interested in the idea that a bacterial product might contribute to uveitis. I was unaware of what A.C. Woods had reported. I was unaware of the trials from the Mayo Clinic, but I was interested in the HLA-B27 relationship, and I was working with Professor McDevitt. In addition to ankylosing spondylitis reactive arthritis that follows Shigella, Salmonella, or Yersinia infection can be and is HLA-B27 related. And what I discovered in trying to induce reactive arthritis in rats is that just the endotoxin component from the cell wall of gram-negative bacteria could be placed into the footpad of a rat to induce a reproducible acute anterior bilateral uveitis. So let's move on to the fourth concept. Does the microbiome contribute to the pathogenesis of ankylosing spondylitis and or acute anterior uveitis? Let me see if I can juxtapose some of the concepts that I've given you for background information. The big question would be, do HLA molecules have an impact on the microbiome? You may know that the HLA system is incredibly polymorphic, meaning that there are many different alleles or alternatives for HLA genes. Most genes in the human body have only a couple of different choices or alleles, such as eye color has very few choices. On the other hand, there are hundreds of choices for even HLA-B27. There's B2701, 02, 05 is the most common, but more than 100. And when you add up the different HLA-A types, B types, C types, DR, DP, DQ, there are millions of possibilities. The theory is that that polymorphism helps to prevent different plagues or bacterial infections eliminating the human race. So we know that HLA molecules affect the response to infections or to antigens. And my colleagues and I have shown that HLA molecules do indeed shape the microbiome. We did this in collaboration with Matthew Brown. The lead author was my deceased collaborator, Mark Asquith. And we reported this in the journal Arthritis and Rheumatology about a year and a half ago. We examined both HLA-B27 and we examined HLA-DR4, which predisposes to rheumatoid arthritis. And we concluded that either allele had a profound impact on the gut microbiome and skewed the bacteria that were present in the microbiome. We also worked with Matt Brown to look at HLA-A29, the HLA molecule that predisposes to birdshot retinochoroidopathy. And we reported about a year ago that indeed A29 also shapes the microbiome. We think that virtually any HLA allele is going to have just enough effect on the immune response that there's going to be a change in the ecosystem, which is represented in our gut. Now, if that ecosystem changes, there are different reasons why we might develop an immune-mediated disease. For example, Rachel Caspi and her group at the National Eye Institute has suggested that there may be mimicry with a bacterial antigen in the gut that could predispose to uveitis. We tend to favor, at least for HLA-B27 related disease, the idea that changing the microbiome causes what's known as a dysbiosis that changes the permeability in the gut and allows bacterial products to get into the lymphatic or the venous circulatory system. So you start off with a genetic predisposition that changes the gut microbiome. There are lots of data to support the idea that patients with ankylosing spondylitis have altered gut permeability. It changes the immune response, or it leads to an immune response if some of those bacterial products end up in the anterior uveal tract or the gut, or the joint or the spine. And that results in the inflammation that we recognize as ankylosing spondylitis and acute anterior uveitis. If indeed these hypotheses are correct, it may be that we can exploit the microbiome for therapy. And indeed, there's been some success in the global transfer of fecal microbiota in diseases such as ulcerative colitis. If we get more sophisticated, I suggest to you that we won't need to transmit or transplant feces. We can transplant specific microbiomes and microbes rather. And it may be that we don't even need to transplant a microbe. Maybe all we need to do is figure out why the gut is permeable and seal off that permeability. Perhaps a drug like sulfasalazine accomplishes something like that. In 2013, Science wrote a commentary about fecal transplants, and they called that the promise of poop. And believe me, although poop is not very pleasant to many people, it may well be the future of immunology, and it holds a great deal of promise for therapy. So what I've tried to show in a very brief period of time is the idea that HLA molecules do indeed impact the microbiome, and that change in the microbiome affects the immune response. And I think it affects the immune response just enough to probably explain why HLA molecules contribute to more than 100 diseases, including birdshot and acute anterior uveitis. These are the people with whom I collaborated over the years, and especially Steve Plank and Mark Asquith were fundamental in the information that I shared. Thank you very much for your attention.
Well, that was a very interesting talk with a lot of promise for the poop in future, a lot of thoughts for us. And with this, we go on to Manfred. And I think Manfred is going to talk about the topic that he loves to talk about. Yeah, Manfred,
Well, I try to do my best. Yes.
Oh,
So,
HLA-B27 typical acute anterior uveitis. Over to you, Manfred.
thanks. So, how can I get rid of this one here? Yes. So we have a little bit time actually, and due to this one, I think it's, let me get rid of this one. Yes. Let me give you two informations about service of the ISG. Number one is you see here our website, and you see probably here the list of all these chapters. Ask advice. So let's suggest you have a patient which is really complicated for you. We can offer you to help you. Go to ask for advice, and then this page will offer. You have to go on this one to find out how it works. You have to find, we have to find out that you're an ophthalmologist or something like this, and that you're co-working with us. And you will then get a template to upload the whole situation of your patient. And then you will get the name of a moderator and send the whole case report to the moderator. He will optimize that case with you and then send it to all members of the ISG. The members have 2 weeks time to come to talk about that case. And then the moderator will give you free for you and the patient, please, our opinion of this situation. Please use that service. That's number one. The second one, which we can offer you, is to, if you go on membership, as you can see here, then this opens here opportunities, and that gives a list of all ISG members with educational suggestions, grants, fellowships, and so on. And it looks like this one. This is the beginning of these tables, depending on the country. It tells you exactly what type of grants, fellowships, probably also residencies are possible in all of the members of the group. That's from Abu Dhabi to the last country, Z, like Cyprus, I don't know. Let's go to my topic. HLA-B27, typical acute anterior uveitis. I think we've heard some wonderful talks about HLA-B27, how important it is. I'm not so sure that this is all, that B27 is really the most important thing to analyze. Well, this is a clinical picture probably from your uveitis practice. You see a lot of patients there with different types of disease. And it may help you now to optimize the diagnosis of uveitis if you can bring in the anatomical type, differentiate them in different anatomical types, acute versus chronic, and then probably also associated disorders, and that may bring the optimal treatment. While we are talking about anterior uveitis here, as you know, as a classical picture here, you see this classical combined inflammation, very classical form. Well, it depends a little bit, actually, how you look on your disease. So, for example, if you think this is a window with the clouds behind, then you're a little bit confused with this picture. But if you think this is probably a window which has on the glass some clouds, and if you open, it has a black, it's dark outside already, then this changes your way of thinking. So with this, I hope I can convince you a little bit. If you use this concept, which I present soon, then you get a quite uniform disease. Let's check the old differentiation of anterior uveitis first. This was to separate HLA-B27 positive from negative patients. We know 40 to 70% of all anterior uveitis are B27 positive. It's more frequent in males. What about females? More frequent in males, and it's more associated to other typical disorders. That's fine. And others, we simply say, well, it could be viral or bacterial, but this is a real problem. Idiopathic, this means we don't know anything, 30 to 60%, and this is one of the real problems. So my suggestion is that you better use the criteria of laterality. Let's analyze what we have available in our patient with anterior uveitis. It could be both eyes. This is quite rare. Probably you find it in 10% of patients only. Simultaneous bilateral, okay? And often in these patients, you find some kind of systemic disorders. If it's always the same eye, think strongly about viral anterior uveitis. This is a classical, very, very typical sign. But then we have the group of changing sites, the so-called ping pong mechanism. And there you have a B27 positive, but also negative acute anterior uveitis. That's what I call typical. So my definition of B27 typical uveitis is a clinically definition. It has to be acute. It has to be unilateral with a changing pattern, and especially very important and very often seen in the patients, you should find preclinical sensations. That could be B27 positive, which are more in men, or negative. And to make it, to conclude this, the treatment for me, if I have such a typical uveitis, is independent if it's B27 positive or negative. So have a look for this patient. It's a 57-year-old male who is B27 positive. Now he comes up with a new recurrence of acute anterior uveitis, and he has preclinical pain behind the eye. And here you see the ciliary body with some swelling here, and that explains the pain. Before you see a lot of cells. So he came on day 1 and he only shows very, very mild cells. Be sure on day 2, day 3, this will be increased to 3+ cells. So this preclinical pain is super characteristic for that type of uveitis. Another thing which is super characteristic is the endothelial precipitates. You know, the typical location are in Art's triangle. It can be centrally or paracentrally. The important point is that it's over the equator, and then it's typical for HSV or VZV. We are not able to really differentiate these things, and I call it endotheliitis. If it's distributed all over the cornea, we call it stellate. This is classically Fuchs. And if it's granulomatous, well, these are granulomatous disorders. Occasionally, I see that also with HSV, but this is actually quite rare, I think. And then you have the classical findings of dust-like precipitates, hardly to see. You really have to work with your slit lamp to see these precipitates, and that's classical for HLA-B27 typical anterior uveitis. Here again, these pictures, granulomatous, dust-like, central, paracentral, highly classical for viral, and here the beautiful picture of Fuchs. Here again, granulomatous, more in Art's triangle. With this one, I would always exclude B27 typical disease. It's so clear. And here, probably you have to look for this one. Do you see here? I think that picture has been seen before. You see some kind of dust-like precipitates. Very difficult to see. It's not centrally. It's more here in Art's triangle, but it's distributed and hardly to see. So why do I try to differentiate this into B27 typical anterior uveitis? It's a more definite diagnosis. It's less idiopathic anterior uveitis at the end. It's more effective. We will publish this very soon. It's more effective treatment. I told you about the treatment already. You should start very early with the treatment. And this is something patients should have steroids already available and start with the first preclinical findings. And it finally, of course, because it's less severe, it ends up with less complications. So what about this patient? You have a 30-year-old patient, ankylosing spondylitis known since 10 years, and he's B27 positive. He has anterior uveitis in one eye since 5 years, increase of intraocular pressure, no clear prodromal signs, which again is not so often seen in these patients. So this is easy to call B27 positive acute anterior uveitis. And I can tell you this was the anterior chamber, and this is definitely not the classical finding we have in this type of uveitis. This is more an infectious one. So let's analyze that case report again. 30-year-old is fine. Ankylosing spondylitis is fine. Since 10 years, okay, they get their recurrences. B27, okay, fine. That's what we expect. Anterior uveitis in one eye without change in 5 years. This is rare. I see that probably one eye for 1 or 2 years, but then it may change the eye. So this is very weird. Increase of ocular tension. We've heard before, the classical finding is actually in acute situation without a long history, of course, that this intraocular pressure goes a little bit more down, not up. Probably after some time, after 10, 20 recurrences, it goes up. No clear prodromal signs. Again, this is, for me, pretty against this B27 disease. So it's probably wrong, B27 acute anterior uveitis. And to be honest, this fits perfect to HSV or zoster, acute anterior uveitis, treatment with adequate antiviral treatment that was highly effective in this patient, and he had actually a combination of ankylosing spondylitis and viral anterior uveitis. You should be able to analyze this one and to differentiate this, definitely. So in conclusion, anterior uveitis clinical findings are more important than HLA type. Use laterality for differentiating anterior uveitis. HLA-B27 typical acute anterior uveitis for me is defined, can be B27 positive or negative. The clinic is more important as these are the changing side, the acuteness, definitely acute, the low pressure, and the prodromal signs. And then immediately start, even if it's in the night, the patient wakes up with his prodromal signs, start with topical steroids probably every 30 to 60 minutes and add some kind of mydriatics. Tell the patient to go to his doctor the next day. When the doctor sees probably very few cells, you're on the right side. Normally, if you're such a disease, quiet disease, there are no cells in. So that's also few cells is suggestive for recurrence. As a consequence, not all B27 positive uveitis are B27 typical AAU. Think about that patient with ankylosing spondylitis and virus. HSV, Fuchs, intermediate uveitis, etc. can be B27 positive, of course, very often. Optimal characterization leads to a better treatment, clear prognosis, and to less idiopathic forms. B27 positive or negative, I think it's okay to differentiate them, yes, but both can be typical AAU. If they are positive, then there is more associated systemic disorders. We heard this from Dennis already, and something very interesting probably has to do with the microbiomes, that only the positive patients really respond to sulfasalazine. The negative ones treated with sulfasalazine may end up even with a higher amount of recurrences. We have a few examples of this one, very weird. Well, this is the recent book we published about intraocular inflammation. And if you work like this, your grading, your typing of uveitis, you probably have more time to relax, probably here on the Cook Islands. That's probably 2 hours flight only from our friends from Australia. Okay, thanks very much for your attention.
Thank you, Manfred. Right now, we all want to be in Australia.
Definitely.
So with this, we move on to the treatment part, and I invite again Peter, and Peter is going to talk about approach to treatment of anterior uveitis. I guess this is very important. We already know now when to start treatment, and once we know how to treat. Over to you, Peter.
Great. Manfred, can you unshare your screen for me? Thank you.
Okay.
Yeah, that's great. Okay, everyone can see me there?
Absolutely, Peter.
Great. Well, look, we're going to start now talking a little about treatment. And basically what I want to do is just go through how I look after a patient with an episode of acute anterior uveitis and how I think about it. So the way I approach these patients is the sort of standard process that we use for all patients with uveitis: a clinical assessment based on a history, careful full examination, and a thorough review of systems. Our clinical assessment, one of the important things is to determine the cause of any visual loss that's present, work out whether it's inflammatory or non-inflammatory. Based on our clinical assessment, we will do selective investigations to try and rule in or rule out specific diagnoses and to help us recognize any sort of a syndrome that might be present. And after all of that, then we can plan our treatment. As I said, clinical assessment is the basis of what we do. History, examination. We often go backwards and forwards, so start to take a history, start to examine the patient. The examination findings help to direct what sort of extra history we might want to take and where to focus our comprehensive review of systems. I often look at my patients' hands, their feet, their joints, their skin, because particularly with anterior uveitis, you can get a lot of clues as to what's going on. And as I mentioned, our aim is to either recognize a syndrome or come up with a sensible differential diagnosis. The thing, as Manfred was saying, is that although there are some useful clinical signs in anterior uveitis, particularly the charatic precipitates and their distribution, a lot of the time the clinical signs of anterior uveitis are fairly non-specific. And whether something is unilateral or whether it's alternating from side to side or whether it is simultaneous bilateral onset is a really useful clue to help direct us as to what some of the likelier causes are. Certainly anterior uveitis can be of variable severity, but it's critically important to remember that you can't say someone has anterior uveitis unless you've examined the fundus and been able to visualize a normal fundus on a dilated exam. Our treatment for uveitis is driven by the threat to vision, the severity of the inflammation, what impact the disease may be having on the patient's quality of life, and whether or not there's an associated systemic disease. These signs of really intense inflammation often are things that make us want to escalate our treatment or to begin systemic therapy earlier than we normally would, and so are important signs to see. That said, however, we know that the vast majority of our patients with anterior uveitis can be controlled with topical therapy. And so certainly my initial therapy is either prednisolone forte or Maxidex drops. And if anyone's got any significant uveitis, I like to use a cycloplegic. I know that there is a lot of shall we say, a wide range of opinion as to whether or not to use cycloplegics or mydriatics. I have a fairly strong belief that we should use cycloplegics, and I'm an atropine user, and I think we'll be able to discuss that further in the Q&A sort of section. There are a number of sort of high-risk patients where we need to be very careful when we are looking after these patients. Obviously, patients with only one eye or one seeing eye. As I mentioned earlier, patients with very severe anterior uveitis, fibrance, and a lot of synechia, bound down pupil, hypopion, and patients with just ongoing severe pain that isn't getting better with intensive topical treatment. Steroid responders can be much more challenging for us to look after. Obviously, patients with inflammatory mediated vision loss, we need to manage their CME and watch their vision. And children behave differently management-wise and in treatment response to adults. And so they're a really specialized group, which we're not going to talk about tonight. This is one of the few papers that I can find that talks about the duration of attacks of acute anterior uveitis. And interestingly, the median duration of an attack was 6 weeks, and the minimum was 3 weeks in this series from Johns Hopkins. But the range was basically from sort of 8 to 12 weeks was the maximum. And I think that's the important number to sort of remember from this paper. So this is a very stylized and certainly shouldn't be taken as absolutely the way to do this, but this is the way I tend to approach someone with a typical episode of acute anterior uveitis. So when I first see them, I want to confirm the diagnosis with my clinical exam. The majority of patients, but by no means all of them, usually have quite intense inflammation, so 3 or 4 plus of cells. I must see a normal fundus, so I'll always examine the fundus, and then I'll typically start them on hourly drops and atropine. I like to see my patients after about 3 to 5 days. Again, see how they're going, make sure their pain's getting better, their vision's okay, there's no new synechiae and synechiae that are there are broken, there isn't fibrin or a hypopion. And I always look at the fundus again. Twice now I've seen patients who, when I first saw them, had a normal fundus and later, within a few days, had developed severe viral retinitis, which wasn't obvious clinically when I first saw them. So I'm paranoid and like to check the fundus again. If things aren't going well, this is when I would consider increasing treatment, subconjunctival steroids or oral steroids. If I assume the patient's doing well, I will continue them on hourly drops and atropine and see them in 2 weeks. By 2 weeks, I'd expect that the inflammation is much better, hopefully controlled. I'll check the vision and the pressure, and if things are going well, start to cut down the drops. So they've had 2 weeks of hourly drops, then they'll have 2 weeks of second hourly, 2 weeks of 6 times a day, and I'll see them in a month. I'd expect there to be no cells, and assuming vision and pressure are okay, I'll then slowly dial back their treatment by dropping a drop a week until they're on 0. And I like to review the patient when they're off drops to make sure that their episode has resolved, that everything's normal, and then I'll discharge them from care, but make sure I tell them what to do if they have a recurrence, if their symptoms come back. As Manfred was talking about, these patients often know when they're getting an attack, and it's common for them to get a recurrence. So I'll usually I'll give them a prescription so they can get drops to start immediately if they have more trouble. So as I said, the majority of our patients we can control with topical therapy. Severe inflammation or cystoid, we'll often use periocular steroids, typically subconjunctival steroids here. Severe inflammation, these patients are typically relatively young, they're usually in good health, they can tolerate a course of systemic steroids, so I have a low threshold to introducing oral steroids for these patients. We're not really going to focus on these patient groups tonight, but we'll often use oral methotrexate for frequently recurrent or patients with chronic uveitis. And these days, particularly in B27 related disease, we'll try and access a biologic, a TNF blocker for severe recurrent disease. We know that regional steroid therapy works very well, and I won't sort of belabor that, but we use subconjunctival steroids, periocular steroids for CME, and Ozurdex or intravitreal steroids for severe CME, obviously when you haven't got a steroid responder. As I mentioned, I have a low threshold for using systemic steroids. So if I can't control the uveitis on maximum topical therapy, they've got progressing signs, they've got continuing pain, or they've got very severe inflammation at presentation, or they've got cystoid macular edema, they're the times that I tend to pretty much very rapidly escalate my therapy to include oral steroids. And certainly if any patients with anterior uveitis that's active or recently active need surgery, then I'll usually cover them with oral steroids. I have on occasion used intracameral TPA to treat fibrin veils, and this works incredibly well. It's not necessarily available all over the place, but I find it's very useful in this situation. It often just sort of kickstarts control of the uveitis. So as I said, I think this is my approach to treating these patients. It works well in the vast majority of patients. Topical therapy is the best treatment for most of our patients, but there are a range of other therapies that we do need on occasion. So, in summary, acute anterior uveitis is a relatively benign disease that's commonly associated with systemic diseases. And once you get the attack under control, you can discharge those patients from care as long as there's a plan that the patient understands if they have a recurrence. Patients with chronic anterior uveitis are the ones that really we can't let go and need to watch carefully because they have lots of complications. So look, I'll stop there and stop sharing my screen and hand you back to Vishali. Thank you.
Thank you, Peter. And with this, we move on to the last talk of the session by Dennis. And Dennis is going to tell us about neo-treatment. So I see in question box, a lot of people are asking about, like, if you can't give steroids, what next? So maybe we will get quite a few answers from Dennis. Over to you, Dennis.
Thank you, Vishali. You can see my screen okay?
Not yet.
Oh, no.
That's okay. We'll see.
Running screen.
Yes. Now we can.
Okay.
Yes. It has to go to slideshow. Yeah, perfect.
Okay, thank you, Vishali. I'd like to discuss some of the new or emerging therapies for the severe and non-infectious forms of anterior uveitis. Peter's given a very good overview of the approach to the management of anterior uveitis, and it's only a small percentage of patients with anterior uveitis that require systemic medication. And I suppose that's what my role is in the clinic that we do together. So it's often worthwhile reflecting on what are the aims of treatment in patients with uveitis. These include the preservation of vision, the relief of symptoms, of the pain, the photophobia, etc., that Peter's already outlined. It's also important to control the associated systemic disease, mainly the rheumatic diseases that we've discussed this evening. The prevention of uveitis complications, and something that I think is just as important, particularly when we're using systemic therapy, is the prevention of treatment complications. Not only the prevention, but this also requires an anticipation of the problems that may arise with these medications, and I'll discuss these as well. If one reflects back about a decade ago when this publication was produced in the Journal of Ophthalmology, looking at the systemic therapies in people with non-infectious uveitis, and this included anterior, intermediate, posterior, and panuveitis, you can see that we're still using a large number of the drugs that were used 10 years ago, although things have changed significantly in the period of time. There's still reliance on steroids for induction of therapy, and none of the alternatives to steroids work as quickly and as effectively as at inducing remission and controlling the inflammatory response as do steroids. However, this comes at a price, which we'll discuss in a second. There are DMARDs that were used then and are probably still used extensively now. Cyclosporin was one of the agents, as were alkylating agents we used previously. We don't use, or in our clinic, we don't use alkylating agents or cyclosporin anymore. And mycophenolate and mofetil has taken their place. And we use that more frequently with methotrexate in most patients with severe disease, as I'll discuss. Non-steroidal anti-inflammatories also are limited use in this disease. what was interesting then and is still applicable is that combination therapies are often still used in people with severe anterior uveitis. One of the problems that we face, and certainly was a major problem previously, was the use of glucocorticoids. These are the group of drugs have the highest morbidity and mortality of any drug commonly used in medicine. The side effects of steroids are protein and common, they're predictable, they're potentially preventable, and overall they're actually poorly managed. Here's an example of a young boy with JIA treated with high-dose steroids, his younger brother, so showing his growth retardation, the side effects of osteoporosis with collapsed vertebra, hirsutism, acne, not to mention the other metabolic diseases and hypertension that follow the use of steroid therapy. So there have been emerging treatment paradigms that occurred over the last decade in terms of the management of systemic therapy in patients with uveitis and severe anterior uveitis included. One of the major changes is an attempt to minimize or avoid high-dose long-term corticosteroid use. Obviously, we should treat the associated systemic disease, and as we've already indicated, treating the associated systemic disease will often lead to control of the ocular inflammatory response. Peter's already indicated how topical therapy is used before local therapy, and then systemic therapy only follows if these are ineffective. We've followed a policy of early introduction of steroid-sparing therapies, which I'll outline shortly. And also it's important now, and one of the major focuses in rheumatology and also more recently in ophthalmology, is treat to a target with controlling inflammation, preserving visual acuity, intraocular pressure, and cystoid macular edema. In the case of diseases that are relapsing and following a relapsing remitting pattern, we would now consider the need for a disease prevention strategy, and I'll discuss this as we use both methotrexate and we previously used azathioprine, both of which have been shown to be effective in preventing relapses in severe recurrent anterior uveitis. And it's also important to consider a combination of drugs and emerging therapies when other strategies fail. So what are the indications for systemic therapy in patients with non-infectious uveitis or anterior uveitis? These include active systemic disease that requires systemic therapy, severe acute anterior uveitis that fails to respond to topical and local steroid therapy, severe recurrent anterior uveitis, and people who are steroid responders with severe acute recurrent or chronic uveitis, and of course vision-threatening chronic anterior uveitis, all of which may require systemic immunosuppressive therapy and steroid therapy. There have been several reviews of systemic therapy in uveitis, including anterior uveitis, and they basically have shown that there's been moderate success with most steroid-sparing immunosuppressive agents, and this has been demonstrated in the SITE study, the MUS study, Proctor study, and the recent systemic review of therapy of systemic therapy in uveitis, showing that in fact systemic therapy is probably superior to steroid implants in some groups of patients. The response rate to systemic immunosuppressive therapies tends to improve over a 12-month period in these patients. And it should always be remembered that the ocular inflammatory response is not controlled in up to 30 to 40% of patients. These studies have also highlighted that there's inadequate steroid sparing in a large proportion of patients, over 40% of people. And overall, ophthalmologists and physicians are poorly adherent to the guidelines for therapy in the case of severe anterior and posterior uveitis for that matter as well. This is our approach to the management of severe non-infectious anterior uveitis that may require systemic therapy. On the right-hand side of the slide, you can see that if the person has a systemic disease, then we would treat the systemic disease. If this responds, then we would consider whether there's need for maintenance therapy or relapse prevention in this group of people. If there's continuous inflammation, we try to induce remission with a short course of systemic steroids, or as Peter's outlined, aggressive topical or steroid injections or implants. We'll then try to introduce steroid-sparing drugs. The common ones we now use are methotrexate and mycophenolate. And less so, we'd use azathioprine or cilazopirin. Cilazopirin we've used mainly associated with inflammatory bowel disease. If one of these steroid-sparing regimen fails, we would either change the DMARD, or more commonly these days, we'd go straight to a biological agent, such as anti-TNFs and occasionally interferon. Our main experience with interferons, however, with posterior uveitis, not anterior uveitis. It's interesting to reflect, in fact, that our approach to the treatment of severe anterior uveitis is very similar to the approach that the rheumatologists take to the treatment of the spondyloarthropathies, whether this is a peripheral spondyloarthropathy, which may be managed initially with a DMARD or a biological. And then if this is ineffective, then these people would be then treated with a TNF inhibitor or an IL-17 inhibitor. If there's axial disease or axial sacroiliitis or spondylitis, a similar approach, initially with non-steroidals or a DMARD, and then if there's failure of this, to a TNF inhibitor. They would then, if there was a non-response after a 3 to 6 month trial of that particular TNF inhibitor, then they were switched between the TNF inhibitors, and there's now 5 TNF inhibitors available to allow one to change from one to the other. And if this fails, then a combination of therapy would be introduced as well, with a DMARD and a TNF inhibitor. There are now many studies showing the biologicals are actually quite effective in the management and treatment of severe anterior uveitis, particularly the TNF inhibitors. They're effective in the spondyloarthropathies, inflammatory bowel disease, and JIA-associated uveitis. There's compelling evidence for their efficacy in Behçet syndrome. There's about an 80% early response rate to TNF inhibitors in patients with severe uveitis. These drugs do not induce a long-term remission, and unfortunately, stopping the drug usually leads to a relapse of the disease in severe, recurrent, and chronic uveitis. With the advent of biosimilars, there's going to be an increasing range of drugs available to us to use in the clinics to control patients with these severe diseases. As of yet, we haven't really ascertained what is the total duration of therapy that one needs to keep a person on a biological for example, to control the disease. And this requires regular review and reflection, and we usually like to have no active disease for 6 months before trying to withdraw their TNF inhibitor. Fortunately, these TNF inhibitors and many of the other biologicals, in fact, have acceptable side effect profile. They're usually suggested not to be used in pregnancy. However, our experience now with adalimumab in particular has shown that it is safe to use in pregnancy, and we've had several patients recently who've had children whilst on adalimumab. This is a study that's interesting to reflect on now. It's over a decade since infliximab was first used and compared to cyclosporin in decreasing the frequency of uveitis cases, and showing that infliximab, in fact, was more effective than cyclosporin in this pursuit. More recently, there's been studies looking at patients with ankylosing spondylitis who have recurrent or early onset or anterior uveitis associated with their seronegative arthritis. And as you can see from this graph, that over time, the infliximab and even more so adalimumab are quite effective in decreasing the uveitis incidence rate over time. Etanercept is not as effective in this sphere, and most of us would now use adalimumab or infliximab as a choice for biologicals in the management of patients with severe anterior uveitis, particularly that associated with a seronegative arthritis or with Behçet syndrome. Our experience also looking at the use of adalimumab in terms of the time to treatment failure, using the same regime that was used in the Visual 1 and the Visual 2 study, showing that in fact, whether a person had an active disease at baseline or inactive disease at baseline, there is a failure rate, but this is an acceptable failure rate consistent with that used with high-dose steroid therapy and other immunosuppressive therapies in this disease. If one is going to use a biological, then you need to be cognizant of the side effects of this and the contraindications to the use of these agents. And pre-treatment assessment is really important in this context. Infections, particularly TB and chronic viral infections, are a major problem in patients who are immunosuppressed, particularly with biologicals. The presence of heart failure and a demyelinating disease is a contraindication to using these biologicals. It's often, in fact, necessary to do an MRI of the brain in people, particularly who have intermediate uveitis, and there's an overlap with MS and other demyelinating diseases. Similarly, malignancy is a contraindication in most cases to the use of these diseases. They can be combined with immunosuppressive therapy, and in fact, we often combine the use of a TNF inhibitor with methotrexate to control patients with severe uveitis. I've already mentioned that pregnancy and lactation were considered contraindications, although at least for pregnancy, adalimumab seems to be acceptable. It's important to consider immunization in people who are going to be immunosuppressed. Unfortunately, we're not often given the option of being able to immunize a person before they commence biological therapies, in which case it's important to remember that if they need immunization, this should be done with a non-live vaccine, and live vaccines should be avoided in people on biological and heavy immunosuppressive therapy. In terms of what targets for therapy are available for people with severe uveitis, particularly HLA-B27 anterior uveitis, in terms of other agents besides the TNF inhibitors, IL-17 has been trialed in 3 cohorts now and found to be not as effective as TNF inhibitors. IL-23 is probably effective in psoriatic arthritis and psoriasis, and its efficacy in uveitis is yet to be fully established. JAK inhibitors are becoming commonly used now to the treatment of seronegative arthritis, and there are trials looking at their efficacy now in patients with severe anterior uveitis and other forms of uveitis. Jim Rosenbaum's already indicated that there is potential to manipulate the gastrointestinal tract microbiome to bring about resolution of uveitis, and this is a potentially exciting area that's yet to be shown to be effective in the management of these diseases. Another agent, the phosphodiesterase inhibitors, such as apremilast, also may be effective in this disease, and they've been tried in the seronegative arthritis. There's potential for gene therapy, and I've already mentioned some of the new genes that have been described and associated with this, and whether or not gene silencing, as has been tried in experimental animal models, and may be effective is yet to be realized. And of course, novel drug delivery systems are another potential development, and this would require a lecture on its own to look at these different ways of delivering drugs to the eye to bring about a resolution of the inflammatory response. So what I've tried to do in this brief overview is outline some of the new emerging therapies for uveitis and emphasize the importance of basically trying to control the inflammatory response early using drugs alternative to steroids to control the inflammatory response. Thank you for your attention.
Thanks very much. Beautiful talk.
So Manfred is going to take over the discussion now.
Yeah, I think we really have some time. That's good. So there are a lot of points to discuss, but I think the most important ones for our listeners should be the treatment. And when I see the frequent questions and answers, there are a few things regarding the treatment which we should probably discuss a little bit. Number one is how to avoid synechia. Atropine is the Australian approach. I think we use it only if we have synechia already and want to break the synechia. Then, of course, we also would immediately use atropine plus some other of these drugs, probably. What is your experience? Well, let me ask you a little bit different way. Patient comes in with a very mild situation with a few cells, but with already few synechia. You've seen the patient before. These are new synechia, probably 1 or 2 days old. You would immediately go to atropine?
Yep. Yep. If you want to break synechia, you've basically got to dilate the pupil. The problem I have with non-atropine-like drugs, we can't get home atropine anymore, but in an inflamed eye, drugs like tropicamide and cyclopentalate tend to only sort of moderately dilate the pupil. And so what tends to happen is that the posterior surface of the iris has that zone of maximum application to the anterior surface of the lens. So if as the pupil expands and contracts, the zone of iris that lies on the anterior surface of the lens changes. When the pupil's widely dilated, or as dilated as you possibly can get it, there's minimal apposition. When the pupil's small, there's relatively minimal apposition. When the pupil is mid-dilated, which is what happens with non-cycloplegic drugs in a hot, inflamed eye, is that you're just maximizing that apposition of the two layers. So in my view, setting the scene for synechia. So I think the decision is either the eye doesn't need dilatation, or it does. And if it does, I'll give them atropine drops. It's safe, it's effective. You know that you've done the best that you can for trying to break the synechia and minimize any further synechial formation. Obviously, the key thing is get on top of the inflammation. It's no good just using atropine. You've got to pour in the topical steroids. And, even if the inflammation's mild, as you were saying, Manfred, it often progresses and becomes more obvious for 7 to 10 days after the onset of the uveitis if you don't treat it, just as you showed with that beautiful, UBM image of the ciliary body edema and swelling. So I treat those eyes very aggressively.
By the way, this ciliary body came from Vishali, please.
Okay.
Thanks, Vishali. Thanks so much. I think something we have to be aware of, this is a super dynamic disease. Yes. Yep. People feel the first. Probably on the next day, we will see the first very few cells. Send these people, well, some people get sent home without any treatment. They get told there is nothing, there's a lot of confusion, sorry about that. Next day, people come back with hypopyon already, yes? So this is really something you must avoid as a doctor of ophthalmology, please, as a uveitis specialist. These people feel something and you should treat them. I would always give mydriatics in such a situation because I feel that on the next day it may explode. If I see these very, very early, it's fine. And I probably can give non-atropine. But of course, if you have already missed the first few days, situation may be a disaster. I agree. Then probably atropine may be a nice one. You have to break these synechia in any way, definitely. There's no way to remove, to leave synechia. And of course, I think you all agree to remove these by small surgery is forget it. Yes, they form immediately again.
One of the people online asked a question in the Q&A about using midrocaine, and if you can get midrocaine, that's fantastic for getting rid of synechia. But sadly, that hasn't been available in Australia for decades. I know people have used dilute subconjunctival injections of adrenaline and all sorts of different things. I haven't tried that, but there are a whole lot of different therapies people have tried to break synechia.
Okay, we now come to the next step, non-steroidal anti-inflammatory drugs. What is your impression about this? Do you use them? If you use them, do you use them prophylactically, as I do? Or do you add them to your systemic or topical treatment when it's active?
I'm happy to be the first person to answer. I don't use it often in acute anterior uveitis. I will add it in if the patient develops cystoid macular edema. I helped some colleagues in Melbourne, Tony Hall, do a trial where we did an RCT to try and look at whether topical NSAIDs would decrease the rate of recurrence of AAU, and in this relatively small study it made no difference. So I don't do that routinely, but I have done it in some patients. I'm not sure how effective a treatment it is. Dennis, didn't you use some, we did a trial with oral NSAIDs?
Yeah, I was going to say we have tried that. I mean, it probably has a role in people with associated seronegative arthritis, because we usually use that as a first-line treatment in that context. If you conclude silazapirene as a non-steroidal anti-inflammatory, yes, it's helpful in inflammatory bowel disease and recurrent uveitis. Yeah. But overall, except, for example, in some cases with macular edema, it's just not something we use commonly.
Yeah.
Okay, that's, I think, the point to, sorry?
I was just going to say, Manfred, I think your point about using silazapirene is a really good one. And Dennis and I have used that in a number of patients very effectively as
in B27 positive people, and not in negative. Yes. So that probably has to do with the microbiome. I think it was even mentioned by Jim. A very interesting point. Yes.
Yeah.
But let's probably go on before we go to the step ladder of treatment. Macular edema. I think that's a very, very, very important point. Macular edema, in my opinion, is a phenomenon which starts in these patients which are already under control. So let's say with 1+ cells, after 2, probably even 4 weeks, suddenly they come back with a drop of vision and you see massive macular edema. Would you agree to this?
Yep. Yeah.
It's not an acute thing.
No, it doesn't. It's not there at the beginning.
Association. And then, sorry?
Vishali, sorry?
It's in the acute phase most of the time. You are so busy treating the acute one that you are not really doing scans to look for it. But it starts coming in 1 week, a very mild one. And what we have documented is, even if it is present on OCT and angio-OCT and FAST in the very acute phase, it just goes away in 3-4 days and nobody even notices it. When it persists, say at 2 weeks or so, that is the time when your uveitis is quiet and you are able to scan better. Those are the persisters, but even in the acute phase, many a time it just quickly disappears.
Well, then let's discuss a little bit the treatment of this persistent macular edema, which can be really horrible high. Yeah. So I think I've learned from Carlos from Moorfields that in this rare situation, topical steroids are highly effective. I really don't know why. I tried it a few times. It is for me. So I use it really very often. I think, Peter, you have your own ideas about that one. How often do you use it?
Sorry, use orbital floor steroids, did you say?
Topical steroids.
That's always the first treatment to try. I think.
No, in macular edema, sorry. If you have macular edema.
I'll if, as you say, my experience is that it comes back usually about week 4 or so. So I will bump up the topical steroids, add in a topical NSAID, and see what happens for a couple of weeks. And if it's clearly not responding, that's when I'll escalate treatment. Yeah. And usually use orbital floor steroids as my next treatment.
Okay. I think and then it responds nicely. But now let's talk a little bit about the use of steroids. Of course, topical steroids is the starting point. I mentioned I would use it after the first symptoms of the patient. My patients have this at home, and I tell them, take your bottle with you wherever you are if you travel. Yes, it's not enough to have a recipe or something like this one, because then probably it's a Sunday and they don't know where the next pharmacy is and so on. They should have it unopened with Mydriatics or something like that, and then really exactly to start. Then I think we agree topical steroids is number 1. the point is, when do we switch to systemic steroid? I would suggest if you have if you see the patient on day 3 or 4 with massive cells, probably even without hypopyon, then topical treatment may be not sufficient anymore after such a time. And it's a minute before forming of hypopyon or fibrin and things like that. And in that situation, I would probably start subconjunctival or systemic steroids in addition to topical steroids. But I do not expect that after 3-4 days of active disease, topical steroids are still sufficient.
Manfred, in our experience, many times the topical steroids which are prescribed are not in adequate frequency, either from patient point of view or even if it is started elsewhere. They just use it 6 times, 7 times a day. And before starting systemic steroids, I would just make sure that their topical steroids are in adequate frequency.
Yeah.
But when they are adequate, you still go on with systemic.
No, I agree with you. 3rd or 4th day, when no response, we would add systemic.
Yeah, because those patients are usually in pain and they're miserable and they're unhappy. Yeah. The one thing sometimes I've tried, Manfred, in that situation is I usually start people on hourly drops. But you can do sort of hyper intensive topical therapy where you get the patient to use the steroid drops every minute for 5 minutes every hour. And that's horrible torture for the patient. But, in 2 or 3 days, that can sometimes get on top of severe anterior uveitis and stop the patient needing to go on to oral steroids. But as I said in my talk, I also have a low threshold for oral steroids. I mean, these patients are young and usually have minimal other comorbidities.
And they need long-term steroids. Systemic steroids, they don't need a very long term.
No, definitely is. Any role for subconjunctival steroids?
I don't use.
Yeah. By the time they see me, they've either had them or it's too late.
Well, when we see hypopyon, it's an indication for me to use subconjunctival, and I expect then per day a reduction for 50% of the hypopyon.
Yeah.
And that for me is more effective than systemic in such a situation. I wonder why, but for systemic may take 1 week to see a real good response.
Yeah.
I'm surprised of this one, even if you have 80 to 100 milligrams.
A word of caution, inject subconj or subtenon only if you are 100% sure it is HLA-B27 negative, because sometimes virals and others may be confused.
Well, we talked a little bit about the differential diagnosis. You have to keep that in mind. Yeah.
Because there is severe inflammation, don't do it unless you're very sure that it's
Yeah, I think that's a really important point for Charlie. I mean, you can always stop oral steroids. You can't dig out subconj. No.
Yeah.
Well, then if you have a situation that you want to switch, how long do you wait with your systemic steroids to get a good response before you switch to immunosuppressive treatment?
Long enough.
Very good.
Manfred, there's no studies giving us guidance in this area. It really is a problem because we're all cognizant of the side effects of steroids, but we still realize it's the only thing that induces remission and has a prompt anti-inflammatory effect in people with severe disease. But I think we need to be cognizant of a timeline that you'd say, look, we're not going to keep this beyond a month, for example, at high dose under any circumstance. Then you have to make a decision to change to another form of therapy.
Would you agree to this one? The patient goes better, becomes better with systemic steroids, wonderful, but you're aware that there is quite a high flare-up, which results in some bad complications. That is a situation that after reducing your steroids step by step to start your immunosuppression. Would you agree to this one?
Yeah, I think you should be looking to spare the steroids at every opportunity. You can assess on your history of the patient whether they're in a high risk group or not. For example, they're hypertensive, got heart disease, got osteopenia, all those other cofactors that are going to make them or diabetic or going to make them much worse on a high steroid regimen. So there are other factors outside the eye that are going to influence how long you keep a person on steroids and how early you're going to introduce another form of therapy.
But then when you are not effective with your next step of treatment, immunosuppression, you're ending up with biologics. And at least in Germany, they are not on label if you're treating anterior uveitis. They are on label for posterior uveitis. And I think in this regard, you really have to contact your rheumatologist. And it's always good for your patient, more or less good, if he has some kind of ankylosing spondylitis. It's very, very helpful for all participants.
Yeah, sometimes those patients have a lot of vitreous inflammation as well. Manfred, it's surprising.
Okay.
And sacroiliitis on their MRI, the sacroiliac joints, yes.
To be honest, in this situation, I would even call macular edema, which is definitely a complication of uveitis, of all types of uveitis.
Posterior
I would
uveitis.
call it as a sign of uveitis of the posterior segment, and then it may work.
Correct.
Yes.
Yeah.
One more point. In case I have a severe macular edema in this situation, I personally would prefer tocilizumab compared to adalimumab, because in my hands, it's much more effective if you have macular edema than adalimumab. What's your opinion about that one? If I have a choice between both.
Yeah, that's an anecdotal sort of response, I suppose. There's no head-to-head study that I'm aware of comparing those two. Manfred, is the problem. Again, tocilizumab is actually quite effective in JIA, for example, and in other systemic diseases. But again, no one's done a head-to-head to show that it's better for macular edema. And the problem with macular edema is that, as we've already discussed, it comes and goes with different forms of therapy anyway. So the long-term prognosis, I don't think can be predicted on whether you use tocilizumab or adalimumab, which is what we would normally use anyway. We don't use tocilizumab for patients with B27 anterior uveitis.
These patients, once you have managed the acute episode, you can maintain them on sulfasalazine and a reduced dose of topical steroids only. Many of them may not even need disease-modifying agents. At least that's our experience.
Well, actually, I do not need normally topical steroids if it's quiet. I don't use that one for probably NSAIDs, but normally the clinic is so easy, for these people to realize it's going on. And I think with 1 or 2 drops, you won't be able to really keep it down for the next ones.
Yeah, but I think Vishal is making a very good point also, and you've indicated as well that the biologicals aren't readily available for anterior uveitis unless you've got a friendly rheumatologist or a drug company that wants to sponsor it, whereas salazapine and methotrexate are cheap. They're cheap, they're readily available, and they're effective. So we often use certainly methotrexate with a biological in the people with severe uveitis.
So I think we are over time now, 3 o'clock in Germany. Any final comments? Something we missed until now from anyone from you? Otherwise.
Could I make a comment about what you were saying about B27 uveitis, Manfred? I think what you made a very good point, and the point of it is that B27 occurs in 8% of Caucasians, but it occurs in no Aborigines or Bantus, and it occurs in 40% of Inuits. So the frequency of B27 in the community varies considerably across populations. And if 8% of the population have B27, they can get other diseases besides B27-related diseases, including uveitis. So just because a person is B27 doesn't mean that their disease is associated with HLA-B27.
Yeah, thanks a lot. It's good. Okay, so if there is nothing else,
we've got plenty of Q&A
we
questions to
still,
answer, Manfred.
so let me close that meeting. Thanks very much for listening. We will try to go over all of your 27 open questions as it is in the moment. Thanks for participating, for asking a lot of good, really good questions, to starting a good controversy here. And we'll meet probably again in a month, then I think with another challenging topic of viral induced uveitis. Have a beautiful weekend. Bye.
Right. Thanks, everybody. It's been good fun.
Bye.
Bye.
So let's check.